I guess let's get started with the warm-up. So the warm-up, again, this is all stuff we went over on Thursday and stuff that will likely show up this coming Thursday. All right, so the first question was about phototransduction and the rods and cones, which is a little convoluted. So it says, which of the following is true when light hits a rod photoreceptor cell? So again, one of the things that's slightly counterintuitive is that the phototransduction actually makes the voltage go down when light hits. We have the dark current. It's more positive in the dark and then becomes more negative in the light. We got 2/3 of people got that one. Right here. Yeah, so cell membrane voltage goes down, and that's happening because the cyclic GMP levels are going down because part of that second messenger pathway is turning on phosphodiesterase, which is breaking down the cyclic GMP, which closes those little sodium channels, which makes the voltage drop. And cisretinol converts to transretinal. That's the thing that starts the whole thing. So again, this is that whole process. Like, how do we go from light to an electrical signal? If there's like some question about, how do we do phototransduction? This is at the core of how we convert light into an electrical signal. Semicircular canals, we talked about the inner ear having these equilibrium senses. Those 3 semicircular canals detect the 3 axes of head rotation. So it's not gravity. What are the organs or the parts of the inner ear that tell you the direction of gravity?
Is it the utricle and the saccule?
Exactly, the utricle and the saccule. Those are the ones that have the little otoliths, those little calcium carbonate stones that will get pulled depending on the direction of gravity. And as they get pulled, they'll bend the hair bundles on the hair cells, which sends a message to the brain about which way gravity is pulling. Actual sensory transduction in hearing, most people got it, is the hair cells in the organ of Corti. Those are the things that are actually converting the actual vibration, the bending, the motion into an electrical signal. Tympanic membrane is the thing that originally vibrates in response to the air vibrations, gets transmitted through the little ossicles to the oval window. The oval window then transmits the vibration into the fluid of the inner ear. In the inner ear, you've got the vibration kind of going up the vestibular duct, back out the tympanic duct. Helicotrema at the apex of all that, but it's the hair cells in that organ of Corti, which is in that cochlear duct, that actually do the conversion of the vibration into an electrical signal. How is air pressure in the middle ear equalized with the outside? Most people got this. That's that eustachian tube. You've got that little connector between your middle ear and the back of your throat, the nasopharynx there. So you can equalize the pressure. So the inside of the middle ear will be the same pressure as the outside, so your eardrum doesn't like implode or explode. This one, a lot of people did not get. Axons that make up the optic nerve from the right eye project to. So this one, if you remember, there is lateralization in vision, but it's for your visual hemifield, like either the left half of your visual field or the right half of your visual field. And each eye sees both of those. So we talked about that optic chiasma. Like the left side of each eyeball is sending its information to one side of the brain. The right half of each retina is sending its information to the other side of the brain. So each eye actually sends information to both hemispheres. So both hemispheres are getting kind of two eyes versions of that half of the visual field, and that's how you're calculating, depth perception and all that. Are there any questions about material that we've gone over up to this point? All right, so to finish up the nervous system, we're going to do motor. And motor is going to be, somatic motor, the voluntary, and then autonomic motor, the kind of smooth muscle and glands and stuff like that. So let's do that. And then actually, once we're done with that, I'll give you a very explicit boundary where here we're stopping with the material that will be on the test for Thursday, but then we will continue on and introduce the muscles because that's the next topic. And it's a great segue going from the motor to the muscles. And also we just need to keep on rolling. So let's do this. Well, actually, I see chats. Let me double check. Oh, we have so people. So yeah. So you guys can, talk amongst yourselves and figure out, make sure everybody's got a Scantron for Thursday. Okay. All right, so again, kind of giving it all context. It was started out with the CNS versus the PNS. The CNS was just the brain and spinal cord. PNS was going to be sensory and motor. We talked a bunch about sensory. Most of the sensory we've been focusing on have been those exteroreceptors, this receptors about the world around you, like the five senses. But like I've mentioned, and we'll continue to see, there's lots of sensory information coming in from the inside of your body. Everything from the stretch of your blood vessels to give you readouts on blood pressure. We talked about proprioception, giving you a sense of the position and movement of your body with like joint angle receptors and muscle stretch detectors and things like that. There's chemoreceptors inside telling you like levels of carbon dioxide and pH and glucose levels and all that. So lots of sensory inside as well as outside. Motor is what we're going to focus on right now. Again, motor, we talked about being somatic. Motor, somatic motor, this is all the voluntary stuff. And this is operating the muscles that you tend to think about as the muscles that you can move on, consciously, like your biceps and your triceps. And, if you're going to dance around, you're going to be using the somatic motor to operate those muscles to dance around. As opposed to the autonomic. Autonomic. For the involuntary in general and glands. Right? You shine a light in your eye, you're not consciously saying, oh, I must constrict my pupil by constricting the circular muscles in my iris. It just happens automatically. So then again, autonomic has sympathetic and parasympathetic, and we'll look at both of those in way more detail. So let's start with somatic, partly because it's just simpler and more straightforward. So the somatic motor division. All right, so if they have the brain and spinal cord, Because typically the signal, in fact, the signals operating your skeletal muscles are originating where? What's the main part of the cortex that is operating your muscles?
Precentral gyrus.
Yeah, exactly. The precentral gyrus, that primary motor cortex. But then those signals have to get out to your muscles. So let's say this is a skeletal muscle. Oops. Again, like your biceps or your gastrocnemius or your latissimus dorsi or whatever, your deltoid. It's just one of these skeletal muscles. It's one of these muscles that you all have heard of. So we need to be able to send the message out of the CNS to get to this muscle and to activate it, excite it. So in somatic motor division, there is always one neuron that travels from the CNS all the way to the target muscle. So there's some neuron, I'm drawing it in blue here. Got a cell body here in the spinal cord, say, and it's got an axon. And the axon goes all the way to the muscle. And then here we have our neuromuscular junction. So what this means is these are some really long axons, right? You have axons that are leaving your spinal cord and going all the way down to the muscles in your foot. That's, the axons that are operating the muscles in your foot have their cell bodies in your spinal cord. So these are really long neurons. The neuromuscular junction we need to talk about. So the somatic motor neuron always releases acetylcholine. So it's going to be puffing acetylcholine here at the neuromuscular junction. So the neuromuscular junction, this is a synapse. It's just like those synapses we saw in the nervous system, like from one neuron to another, but now the bouton is releasing acetylcholine at the muscle. And then obviously what has to be on the muscle itself at the neuromuscular junction?
A receptor?
Yeah, exactly. Acetylcholine receptors. In the somatic motor system, they are always the subtype that we talked about, which are the nicotinic cholinergic receptors. Does anybody remember the kind of defining qualities of nicotinic cholinergic receptors?
Aren't they always excitatory?
Yeah, exactly. These are the ones that are directly gated. They're always excitatory. Right. That means if you send an action potential down a somatic motor neuron, when that excitation reaches the muscle is going to get excited and contract. A little later, either today or next week, we'll talk more detail about the whole idea of excitation contraction coupling, exactly all the steps involved from an action potential down the motor neuron to the actual physical contraction of the muscle. But for right now, just realizing that there's always this excitatory synapse here. This is always excitatory. If you send a message down the somatic motor neuron, you're going to cause a contraction in the target muscle. This is going to be different than the autonomic, where you have more flexibility, more options. So this is basically all I've got for the somatic motor division. The kind of defining qualities here. You need to know that there's just one neuron, and it starts in the CNS, and it just ends up going all the way to the target muscle. And when it gets there, it's going to release acetylcholine, and the muscle itself will have a nicotinic cholinergic receptor, which is going to be an excitatory synapse then. What else?
I have a question.
Sure.
So would there be pyramidal tracks running from the primary motor cortex down to that cell body in the CNS?
Probably, yeah. Those pyramidal tracks, so they're like those pyramids and stuff, are the ones that are then like going down into there for sure.
Thanks.
Botox can bring this into the real world. People get little Botox shots to like, what is Botox doing?
Is it like blocking the nerve that goes down to the muscle?
So it affects that nerve. What it does is it inhibits the release of acetylcholine. So here I'm saying that the somatic motor neuron releases acetylcholine. Botox will come in and stop that. So there's no acetylcholine being released at the muscle, which means the muscle is not going to be excited, even if the brain is trying to send a message down to it. So people get it shot in little muscles around their face that are causing tension. So everything kind of relaxes. But then, the downside is you lose subtle ability to have like express emotions because, a lot of the non-verbal communication is through little subtle facial things, which now you just look a little flat, but your wrinkles are less. So it's people make that choice. Not that I have judgment. No, I don't know. It's time to move on to the autonomic motor division. So any questions about this before we move? Actually, I should say somatic, operating skeletal muscles. Obviously I'm saying it's voluntary, but it's not always voluntary. We saw with the stretch reflex, patellar tendon reflex. Someone whacks on your patellar tendon, your knee's going to kick out. That's mediated by somatic motor neurons, and you didn't do it on purpose. It was a reflex. So, in general, it's voluntary. The diaphragm is also mediated by somatic motor neurons, right? I can make my, I can voluntarily control my diaphragm, but if I forget to do it, it's going to keep going. I don't suffocate if I forget to breathe. So, it's for the most part voluntary, but don't get too caught up on, but it's not always voluntary in every situation. John?
For the parts of the body that have much more fine motor control, like the hands and the face, I always assumed that meant like more innervation, say more than one nerve going to a muscle, but it sounds like that's not the case. It's just more individual muscles, each one with one nerve going to it.
So it's going to be motor units. So we'll talk about that in more detail. Basically, usually a motor neuron like this actually innervates a bunch of muscle fibers.
Right.
It's called a motor unit. So like in your biceps, it's gonna maybe 50 to 100 muscle fibers, muscle cells will get innervated by an individual somatic motor neuron. Like in your fingers or like in the muscles that move your eyes around, it might only be a few muscle fibers that are activated by a motor neuron. So you have much more fine control. You can just turn on a few more, a few less muscle cells to create a slightly stronger or weaker contraction, as opposed to like your calf muscle, where you can only turn on like 500 more or less at a time, because there's like 500 muscle cells that are operated by that motor neuron that's going to your calf muscle.
Okay, that makes sense.
Yeah. Okie dokie. So now we're gonna finish up the nervous system with the autonomic. And yeah, the motor units, if you were to think, wait, what was that? We're gonna be talking about motor units in much more detail as we talk about muscles. So don't focus on that now. So the autonomic motor division, and this is the one that is operating smooth muscles, changing the diameter of your blood vessels or the diameter of your pupil, or activating your salivary glands to make saliva pour into your mouth or whatever, make your sweat glands start sweating. So I'm going to start with things that are common to both divisions, or to both parts. There's, again, obviously sympathetic and parasympathetic, and then we're going to talk about the things that are specifically just sympathetic versus autonomic. So again, here we have the CNS. When we're looking at this division, there are always two neurons from CNS to the target. So for instance, let's say the target is the heart. We want to either speed up or slow down the heart. There's always going to be two neurons. There's going to be one neuron that starts in the CNS, but then it doesn't go all the way. And then there's going to be a second neuron that then continues and makes it to the target. Does anybody remember what we call collections of neuron cell bodies that are outside of the CNS, that are in the PNS?
Ganglion.
Yeah, exactly. We have a ganglion here. So and where the ganglia are depends on whether you're looking at sympathetic or parasympathetic. We'll talk about that in more detail a little later. This is basically just a relay. There's not a whole lot of, for our purposes, not a lot of interesting stuff happens here. This is basically acetylcholine to nicotinic cholinergic. So basically, when this is excited, it just continues on that side because it's just an excitatory synapse that just relays the message. So don't worry about the detail of this. We're going to spend much more of our effort understanding the connection from the postganglionic neuron to the target itself. Ganglion is just kind of like a relay. All right, so now what we're going to do is compare and contrast sympathetic versus parasympathetic. I'll try to be color coded. I'll do like sympathetic in red because it's kind of the one that's more like fight or flight, parasympathetic I'll do in blue because it's calmer. Again, we've talked about parasympathetic, also kind of the little mnemonic like rest and digest, promotes homeostasis and healing. In general, parasympathetic tends to calm things down, promotes digestive processes, slows down your heart. Sympathetic is that fight or flight. It's the one that speeds up your heart, dilates your bronchial passages so you get more oxygen down, so you can like deal with some emergency. In reality, they're both happening all the time, right? The light gets a little brighter in the room. Parasympathetic is going to constrict the pupil, and the light gets dimmer and we need to dilate the pupils. It's sympathetic. So it's not like one or the other is going, like you're either in couch potato mode or running from a tiger mode. They're constantly, people talk about it like driving with one foot on the gas, one foot on the brake, and you're just constantly kind of adjusting depending what is appropriate for what the body is needing to do in the moment. So sympathetic and parasympathetic. Let me put the brain and the spinal cord back here. Let's get our target. Obviously people have one heart, but in order to make this easier, I'm going to put one heart on either side so we can make this easier to see. So now we'll start talking about ways that they're different. So we talked about how they both have two neurons that go from the CNS to the target. One difference is just where the first neuron originates from. If we're in parasympathetic, we have what's called the craniosacral outflow. The first neuron is either a cranial nerve or it's coming from like the sacral region of the spinal cord. In fact, about 70% of the parasympathetic originates from one particular cranial nerve. In fact, it's the only cranial nerve that leaves the head. If you've been in anatomy, what is the wandering cranial nerve that leaves the head? Wanderer, vagabond.
Is it the vagus nerve?
Yeah, it's the vagus nerve. So maybe like 70% is from cranial nerve 10, which is the vagus nerve. Sympathetic, on the other hand, originates from the thoracic and lumbar regions of the spinal cord. So we call it the thoracolumbar outflow. Right. I mean, this has implications for someone with spinal cord injuries. If you have a spinal cord injury, it's likely to dramatically mess up your sympathetic functioning, because the sympathetic is originating down in this thoracic area, whereas it might have more minimal effects on your parasympathetic functioning, because parasympathetic, the majority of it's not even coming down your spinal cord, it's coming down the vagus nerve, just originating from your brain and coming down the foramen magnum and wandering about in your body. So that's one big difference, just where the first neuron originates from. Another one is just the relative lengths of the different neurons. In the sympathetic, the first neuron is short. Second neuron is long. So you have the ganglia are right near the spine. Again, we're not going to get into the details of the sympathetic chain and all that. That's something we do in anatomy. For right now, just we can say all these sympathetic ganglia are near the spine. And then the second neuron then continues to the target. So we have a short first one and a very long second one to our target. Parasympathetic, it's the opposite. Long first neuron and a short second neuron. In fact, the ganglia are right near, or sometimes even within the walls of the target organ. So the ganglia, we call them terminal ganglia. You know, because they're at the terminal, at the far end of this thing, or sometimes they're intramural ganglia. Meaning within the wall of the target even. So that's another difference. And then the kind of most physiologically relevant is what kind of neurotransmitter is released at the target. So in the sympathetic, you typically release norepinephrine or epinephrine at your target. And I'll say typically, you know, I'll say there's a few exceptions here. Norepinephrine and epinephrine. So epinephrine is, you know, I'll talk about that. In the parasympathetic, they always release acetylcholine. Right, and I should say release, you know, at the target. So the last thing we need to talk about here are the types of receptors, though, on the target. So you might think like, oh, just like the somatic motor neurons, it's releasing acetylcholine. But there's a big difference in that the targets have muscarinic cholinergic receptors. So what does that mean? If the target has a muscarinic cholinergic receptor.
It can be either excitatory or inhibitory.
Yeah, exactly. So when you activate a parasympathetic motor neuron, it depends on the second messenger pathway on the target, whether or not it's actually gonna, like, parasympathetic will excite your glands and your digestive processes to increase activity. Parasympathetic can slow down your heart, though, right? So it might be excitatory or inhibitory, depending on the second messenger pathway that's downstream from that muscarinic cholinergic receptor, as opposed to somatic motor neurons where they're always excitatory. So these parasympathetic motor neurons can be excitatory or inhibitory, which is again different from somatic. In the sympathetic, we've already mentioned we have the alpha 1, alpha 2, beta 1, beta 2 adrenergic receptors. These are second messenger gated, or second messenger mediated. So again, these can also be excitatory or inhibitory, right? It's going to speed up your heart, but it's going to inhibit your digestive processes. You're going to get it. If you're nervous, your heart's going to speed up, but your mouth's going to get really dry because it's going to turn off your salivary glands. So this is the basic thing. There's one last thing we need to talk about. A difference between sympathetic and parasympathetic is sympathetic has this one other trick up its sleeve, in that it can trigger the release of adrenaline by the adrenal medulla. So let me add, this is the kidney. And here's the adrenal gland on the kidney. And it turns out one of the things that this system can do is go and turn on the adrenal medulla to release epinephrine just into the bloodstream. So I'm realizing this is messy, so I'm going to erase this stuff, which you already know. And I will write this more clearly. So epinephrine, also known as adrenaline. So what's the difference of triggering the adrenal gland to, and I'm saying adrenal medulla because the adrenal gland has two parts. The adrenal medulla is the part that makes adrenaline. It's the part that's derived from nervous tissue, as opposed to the adrenal cortex, which we're going to talk about near the end of the semester, that is releasing all sorts of steroid hormones. What is the difference between just releasing epinephrine into the bloodstream versus releasing, norepinephrine or something at the target and at these receptors?
It's a much broader response, and it's going to affect kind of everything. It's systemic.
Yeah, exactly. So this is like a blanket. You just broadcast this ligand everywhere. Right? Normally, if you're operating things just using the neurons, they're going to go to some particular target. You'll speed up the heart, but nothing else is going to get affected. or you'll dilate the pupils, but nothing else going to get affected as long as you have a kind of a direct path. But when you trigger adrenaline in release into the bloodstream, it goes everywhere. And anything that has the appropriate receptor is going to respond. Your pupils will dilate and your heart will speed up and your respiratory passages will dilate and your digestive processes will shut down and you'll start sweating, and all this stuff is just all going to happen at once. Actually, I should not, maybe not sweating, but all this other stuff. So, what's the word we use when we have a signaling molecule that actually gets put into the bloodstream and just goes more global? It's no longer called a neurotransmitter. What do we call it now?
A hormone?
Hormone?
Yeah, it's a hormone. So epinephrine is a hormone. Maybe I should write that out. We'll talk much more about hormones later, but this is kind of our introduction to hormones. It's the same basic idea as neurotransmitters, but instead of being localized to a particular synapse, it's just sent everywhere, and any cell that has the appropriate receptor for that ligand, for that hormone, is going to respond. And the other thing that's interesting about hormones compared to directly activating things is once you release it into the bloodstream, it takes a while to break down and dissipate. So, if you get really scared, it's like, oh my God, a tiger, blah. And it's like, oh no, there isn't a tiger. But it's going to be a few minutes before my heart slows down. Because once you dump that adrenaline, that epinephrine into your bloodstream, it takes a few minutes before it gets all cleared out. So it's really important to remember acetylcholine for parasympathetic, epinephrine, norepinephrine for sympathetic, because a lot of drugs are targeting specifically one or the other side of the autonomic nervous system. So for instance, if you're giving somebody a drug for an asthma attack, you want something that is going to dilate the bronchial passages. What kind of a drug would you give them?
Epinephrine.
So epinephrine could do it, but what's a more kind of generic term for a drug that would bind these kind of receptors?
An agonist?
Yeah, an agonist. So an adrenergic agonist. Like albuterol is like what's in those like inhalers. That's an adrenergic agonist, something that binds and activates the adrenergic receptors. What about if somebody has high blood pressure and you actually want to keep the heart slow? You don't want the heart to speed up. You want to actually block the effect of the sympathetic nervous system. What kind of drug would you give them?
Big ass.
Say it again. I didn't hear.
An antagonist?
Yeah, what kind of an antagonist?
Again, an adrenergic antagonist?
An adrenergic antagonist. So like a really common drug for high blood pressure is like propranolol, which is the beta blockers. It's a beta adrenergic antagonist. It blocks the epinephrine, norepinephrine receptors on the heart and keeps the heart from speeding up, keeps things. We're going to talk about much more detail about why blocking beta adrenergic receptors keeps your blood pressure down. It's both slowing the heart rate as well as reducing the contractility strength of the heart muscle, both which work together to reduce, the cardiac output there. What about if if you go in for an eye exam, they shine a light in your eye. They want to look around inside your eyeball. But normally, if you shine a light in somebody's eye, the pupil gets really small and then it's hard to see what's inside there. So the optometrist or ophthalmologist, whoever, is going to put some drops in your eye that are going to keep your eye from constricting. What kind of drug is in those eye drops that is going to allow you to shine a light in somebody's eye but not have the pupil constrict?
Cholinergic antagonist.
Exactly, a cholinergic antagonist. Those little drops they put in your eye that make your pupils get really wide, those are cholinergic antagonists. They block, even though your brain is saying like constrict, the message never gets to the muscles in your iris to actually make them do it. Okay. So again, a lot of pharmaceuticals are specifically designed to modulate activity of sympathetic or parasympathetic motor stuff. I can also say there are, these subtypes of receptors can be more or less localized in like heart tissue versus lungs or this or that. So, or bronchial passages. So there's some like ability to fine tune what exactly, where in your body the effect is, because there are different subtypes of these adrenergic and cholinergic receptors. For completeness, I said that sympathetic typically releases norepinephrine or epinephrine at the target. There are a few exceptions where it actually releases acetylcholine, but it's not something that becomes much of an issue that we need to worry about. Like in your sweat glands, sympathetic fibers actually release acetylcholine to operate your sweat glands. But in general, you should think about sympathetic as adrenaline and noradrenaline, again, epinephrine, norepinephrine, and parasympathetic as acetylcholine. What else? So parasympathetic and sympathetic tend to be antagonistic, meaning they kind of do the opposites from each other. Parasympathetic will slow down the heart and sympathetic speeds up the heart. Sympathetic will dilate your bronchial passages, but parasympathetic will constrict your bronchial passages. Parasympathetic constricts your pupils, sympathetic dilates your pupils. So usually there's this kind of push and pull, but there's a few exceptions that are, worth mentioning for completeness. Sweat glands are purely sympathetic. So more sympathetic activity, you have more sweat, less sympathetic activity, less sweat. This is why people use skin conductance, like the electrical conductance against your skin, as a measure of sympathetic activity. Like part of the lie detector test, you actually, you have somebody's fingers here, and you can look at, what is the voltage across, the more salt water, the more electricity can go through this person, and that means there's more of a sympathetic activation typically. So, we talk about galvanic skin response. It's basically looking at the electrical conductivity of your skin. The more you sweat, the more salt water, the more electrical conductivity, and you can measure it. It's really trippy. If you have, normally if we were in lab, we would do it. You have somebody hooked up to the machine. We have these in lab. They're simple little machines. And you just kick somebody. And all of, if you're looking at the voltage on their skin, you kick them, and all of a sudden, I shouldn't say voltage, the galvanic, let's just call it galvanic skin response, the conductance. It'll just kind of go up. You'll just see it. Or if you just ask them an embarrassing question like, oh, did you have sex last night or something? All of a sudden, you'll just goes way up. So it's kind of fun to like just see how you don't really, I mean, I guess people who are like spies and have to try to like learn to control this kind of stuff. I think there are people who learn to try to control them. But in general, if something is activating your sympathetic nervous system, it will result in increase in sweat, which is measurable as an increase in skin conductance because of the salt water. Blood vessels are mainly sympathetic. The fibers that control the diameter of your blood vessel by doing the smooth muscle is mainly sympathetic. I mean, if you think about it, you don't really need to antagonize. If you have more excitation, it will constrict the vessel. But if you have less excitation, there's blood with its own pressure pushing it back out. So you don't necessarily need to pooch it out. It will pooch out automatically because of the blood inside with its own blood pressure pushing it out. When we get to the vasomotor reflexes controlling blood pressure, most of those are operated by sympathetic motor neurons. The other thing where the example people talk about where they are not necessarily antagonistic is when parasympathetic and sympathetic work together for sexual response. We'll talk about this more when we get to the reproductive system, but parasympathetic is necessary for erection when a guy is getting aroused. But then at some point, during the kind of peak of sexual excitement, there's a sympathetic reflex, mediated reflex, which is the orgasm and ejaculation. So people talk about them working together towards a common goal there. It's part of the reason why guys sometimes have trouble having an erection if they're like really nervous or something, because you need to have the rest and digest system online to actually get the erection. So that's one place where they're not necessarily fighting each other, they're working together. But in general, you can think about them antagonistic. Let me think about this. I think that's it. So, end of material for exam 2. I wanted to, it just made sense to tie off the nervous system before we have the exam instead of leave this little piece left over. So exam 2 is gonna be, it's gonna cover a lot. It's gonna be everything from where we started about the nervous system in general, the brain and the spinal cord, the peripheral nervous system, the senses. So anything from kind of the end of the last exam to now. Are there any questions about any of this material at this moment?
Yes, I have a question. Okay, I just want to know that if I'm thinking of this right. So cholinergic uses acetylcholine, adrenergic uses norepinephrine and epinephrine. And then like if it's a adrenergic, I can't say it, but if it's an antagonist, so it binds to the receptor and like releases norepinephrine and epi.
No, what's the definition of an antagonist?
Oh,
It blocks reception.
It blocks the receptor. It will bind the receptor, but it blocks it. It keeps the endogenous ligand from getting in there. It would be kind of like you put a bunch of like Play-Doh in somebody's like lock, so now they can't use their key to open up their door and they're locked out of their house. Right? It gets into the receptor, does not activate it. Like the Play-Doh is not going to open the door or unlock the lock. But now the person who actually has the key can't get in the house either because there's something stuck in the lock that you can't stick the key in. That's kind of what an antagonist is doing. So if you have a adrenergic antagonist that is binding the norepinephrine and adrenaline receptors on your heart muscle, then even if your sympathetic nervous system is activating, your heart is not going to speed up. If you have the adrenergic antagonist binding the receptors on your bronchial passages, that would be bad. It would mean like you'd want to make your bronchial passages dilate, but they wouldn't. So those, we don't really use drugs like that. We use drugs that are adrenergic agonists because those are going to bind and actually activate and dilate your bronchial passages and make it easy to breathe if someone's having asthma. So agonists bind and activate if you want to make something, turn it on. Antagonists is if you want to kind of stop it from working.
okay, thank you. I think I was just switching those two.
But yeah, again, as you get further on and into your pharmacology and all that, you're going to see just all sorts of drugs that work by being adrenergic agonists or antagonists or cholinergic agonists or antagonists. And again, specific subtypes even, because the specific subtypes are localized in different areas or play slightly different roles. But that's for another class.